Good afternoon, everyone, and thank you for joining us for this week's National Bleeding Disorders Foundation, formerly National Hemophilia Foundation, Wednesday webinar. My name is Fiona Robinson, and I am the series host. This webinar is being recorded and will be available to the community on the NBDF website, bleeding.org, shortly after the webinar. In today's webinar, we will learn about race, ethnicity, F8 variants, and inhibitor risk, analysis of the My Life, Our Future Hemophilia A database. We encourage you to ask any questions you may have today. You can ask those questions by using the Q&A feature at the bottom of your screen at any time during the webinar. We will monitor these questions, and then following our speakers' presentations, we will put your questions to them. Today, we are joined by Dr. Anwar Ahmed, a faculty member in the Department of Preventive Medicine and Biostatistics, at the Uniformed Services University of the Health Sciences, Bethesda, and by Dr. Kathleen Pratt, Professor and Vice Chair for Research in the Department of Medicine, also at the Uniformed Services University of the Health Sciences in Bethesda. I'd like to thank Drs. Ahmed and Pratt for joining us today, and I'll turn things over now to Dr. Pratt to get us started.
Okay, great. Thank you for the very nice introduction, and thank you especially for the invitation. It's an honor and a privilege to be able to give this presentation for the National Bleeding Disorders Foundation webinar series. So thank you. I'll be describing a project that Dr. Ahmed and I carried out and published last year in Journal Thrombosis and Hemostasis, with a little background and going a little bit outside that publication too. So the title of today's talk is Alloimmune Responses to Coagulation Factor VIII. What do race, ethnicity, and protein conformation have to do with it? We are both government employees. We start with our standard disclaimer. Any opinions, assertions that you hear, jokes, anything are from us and are not the Department of Defense or the U.S. government speaking. I am an inventor on several patents related to factor VIII immunogenicity. Dr. Ahmed has no relevant conflicts to report. So as this audience knows very well, hemophilia A is an X-linked bleeding disorder caused by mutations in the F8 gene that encodes blood coagulation factor VIII. Hemophilia A afflicts about 1 in 5,000 males worldwide, and patients may have a mild, moderate, or severe bleeding phenotype. And the phenotypes are defined by the percent of normal factor VIII clotting activity of their blood, or actually their plasma. And so severe hemophilia A is defined as plasma with less than 1% normal clotting activity, moderate between 1 and 5% normal clotting activity, and mild between 5 and 50% normal factor VIII clotting activity. Importantly, there is no known association, at least that I've come across, between hemophilia A incidence or severity with race, ethnicity, geographic background, whatever. So that's a different subtopic that we'll be going into in a bit. For many years, the standard treatment for hemophilia A was intravenous infusions of the missing protein, therapeutic factor VIII protein. And due to the fairly short half-life of this protein, those infusions were generally 2 to 3 times per week for those receiving prophylactic therapy. Now, we all are aware that the therapeutic landscape has really been revolutionarily transformed in the last few years with many new products available. And I think the most significant one currently is the advent of bispecific antibodies, which are a wonderful therapeutic that greatly decreases the need for multiple intravenous infusions because you're looking at the half-life of an antibody rather than the half-life of factor VIII. Nevertheless, bispecific antibodies do not completely substitute for factor VIII because factor VIII is still absolutely needed in cases where you have a major injury, a trauma, or to support surgeries. So factor VIII is still important, even with the ability to treat patients with bispecific antibodies and other non-factor VIII therapeutics. So here is a standard, somewhat simplified picture of the blood coagulation cascade. And the reason I'm showing this to you is to emphasize the position of factor VIII which basically is connects the extrinsic and intrinsic coagulation pathways. So the extrinsic pathway shown here on the right is the normal pathway where most blood coagulation is initiated. It happens when you get injury to the tissue and subsequent exposure of expression of tissue factor, which binds to factor 7A, and then you get a series of proteolytic activations, eventually leading to thrombin activation and activation and polymerization of fibrinogen to make fibrin, activation of platelets, etc. In other words, platelet plug, fibrin mesh, clotting. The intrinsic pathway is the amplification arm of coagulation, and that's another series of serine protease activities. But these two arms of coagulation come together at when factor 9A converts factor 10 to 10A right prior to the activation of thrombin, and like I just said, fibrin polymerization, etc. Now, when factor 8 is in the mix here, the activity of factor 9A is accelerated by 4 orders of magnitude. So hemophilia patients with severe hemophilia A will be missing that amplification there. And therefore, though their blood will clot some, it doesn't clot fast enough or extensively enough. And so this is just basically to show you the importance of factor 8 in coagulation. So it's a wonderful protein. It's essential for coagulation. Unfortunately, factor 8 is also an unusually immunogenic protein. And among patients who receive prophylactic care, which is most of those in the United States, about 30% of severe hemophilia A patients will develop at some point either transient or persistent anti-factor 8 neutralizing antibodies, which are referred to clinically as inhibitors. And inhibitors are the most severe and costly complication of factor 8 replacement therapy. Standard of care is to treat inhibitors by immune tolerance induction, which is essentially intensive factor 8 treatment until antibody titers hopefully subside and factor 8 treatment can then be provided again. ITI, or immune tolerance induction, however, is extraordinarily expensive. Treatment of a single bleed can be up to $1 million. The good news about ITI is it succeeds about 70% of the time. The bad news is it fails about 30% of the time. So we still need to improve outcomes in particular, especially for the patients who fail immune tolerance induction and then don't have access to effective factor 8 therapy, either prophylactically or as a backup for other non-factor therapies. So mechanistically, why do these inhibitors develop in the first place? Well, if you have severe hemophilia A, then fused factor 8 is a completely foreign protein and your immune system has not seen it before. For factor 8, for patients with mild or moderate hemophilia A, which may be caused by just a very minor change in the protein structure, even a single amino acid difference may or may not provoke neutralizing antibodies. And we'll get into details of that in just a moment. So how do you get from a protein to an antibody response? Well, you need a subset of your white blood cells called CD4 T cells. These are called helper T cells because they help the body make antibodies. And CD4 T cells are needed for initiation of the immune response, for cytokine stimulation that then allows B cells to become antibody-secreting plasma cells. They're also needed for affinity maturation and class switching of antibodies. In other words, increasing the tightness with which the antibodies bind their cognate antigen and also increasing the specificity of those antibodies. So CD4 T cells are essential for all those steps. And here's a very simple cartoon that any of you that have heard me give hemophilia inhibitor talks before have seen this. It's just a very simple cartoon I like. If factor 8 in your circulation encounters an antigen-presenting cell, it can be taken up by that cell, chopped up into its component pieces called peptides. If one or more of these peptides then binds to what's called the MHC class II receptor, it can then be transported to the surface of the antigen-presenting cell. Now, if you have a circulating effector T cell that also recognizes this peptide bound to the MHC, you can get stimulation of the T cell cytokine secretion, leading to plasma cells that secrete antibodies. Now, there's another class of T cell called regulatory T cell that can help dampen down this response. And I'm just mentioning that here. We have active research projects trying to exploit the ability of Tregs or regulatory T cells to damp this down, but that's not the topic of today's talk. So from a broader physiological view, this is just a picture I like from an introductory immunology textbook, reminding us that when an antigen first enters the body through blood, the first major organ it sees is the spleen. So the spleen filters the blood, and immune responses to factor VIII typically will start developing in the spleen. And again, if factor VIII is recognized by antigen-presenting cells there, that's where it all starts. Now let's zoom in quite a bit and look at what's going on a molecular level. So remember I mentioned the MHC class II and the T cell receptor. And in brown here is shown the factor VIII peptide, or peptide from any peptide. And the point of this cartoon is to show that there are grooves in both the T cell receptor and the MHC class II that have a very specific shape. And in order to form the immunological synapse where the T cell receptor is bound to the MHC, you need to have a peptide with side chains that fit very specifically into these grooves. This means that the vast majority of peptides that might be chopped up, whether they're from factor VIII or anything else, will not bind effectively to both the MHC and the T cell receptor. But when you do get this effective binding, and when it's tight enough that it stimulates the T cell, this peptide is then called an epitope. And this is relevant to racial ethnic differences that we're going to get to talking about once I get through this introduction. So T cell epitopes in factor VIII, what are they, where are they, how many are there? We've studied this for a number of years. It's a fascinating topic. We use a number of different assays. One type of assay we use is called an ELISpot. So these circles here that you see are basically wells in a plastic plate, and we've added CD4 T cells from a patient, from a hemophilia patient. And we put different substances in together with these cells. And if you see a spot, that means that is an individual T cell that is secreting cytokines in response to factor VIII. So negative control is unstimulated cells. There's always a few of them spitting out a few cytokines. This patient did respond to factor VIII. Positive control is tetanus. Most of us have been immunized against tetanus. And over here, these LP1, 5, and 6, these are pools of factor VIII peptides that also caused an immune response. So that tells us immediately there's more than one epitope in factor VIII. There's actually going to be more than several, probably. We then carry out more experiments not shown, doing the same assays with individual peptides. And in this showing you here is a couple of individual peptides from the A1 domain of factor VIII that also caused an immune response. We then did additional experiments that are flow cytometry analysis using a molecule called tetramers. We can get into that later with experts if they want to follow up. But well, all you really want to see here is if you have cells in the upper right quadrant, these are T cells that are specifically recognizing these peptides. So two assays, both showing you these two peptides are immunogenic, and then we can pull these peptides out, grow them up in culture to get factor VIII specific T cell clones. And when we get those, that's considered essentially the gold standard of confirming whether a particular peptide is or is not an epitope that's relevant to an immune response. So, new topic, it is related, bear with me. In 1998, Lou Alidort and Donna D. McKelly published an interesting paper just noting that they were seeing higher inhibitor rates in their African-American and Latino patients. And they encouraged the community to prioritize this and develop larger studies, see if this was real, and if so, it would be important to figure out what's going on. So my lab and others have looked at this over the last decade or so, and we asked the questions: do some racial or ethnic groups actually have a greater risk of developing inhibitors, and if so, what is the reason or mechanism? So in 2009, Field et al. Came up with a hypothesis that was eminently testable experimentally. They basically sequenced the factor VIII gene in a large number of individuals, all different races, different ethnicities, and found that there were some sequence variants that were more prevalent in the African-American population than in the white. And so H1, H2, H3, H4, and H5 are different factor VIII sequences. And H stands for haplotype. Haplotype means the identity of several amino acids that have been found to be variable in the population. And you can see that white people, 93% of white people that they sequenced anyway, have H1 haplotype. The remaining 7 have H2, whereas H3, H4, and H5 were found in over 22% of the Black population. So the immediate question was, could these sequence variants correspond to T cell epitopes, which I've just been telling you about for the last 5-10 minutes? So given this sequence variant in the factor VIII sequence, it's clear that a number of hemophilia patients have been infused with sequence mismatched factor VIII, meaning factor VIII that has a different haplotype than the gene encoding factor VIII in their body. So it turns out all recombinant factor VIII products correspond to haplotypes H1 or H2. Those are the white haplotypes. Now, don't overinterpret that. There are plenty of Black people that are H1 and H2 as well, but for simplicity, they're overrepresented in the white population. Plasma-derived factor VIII also has plenty of H1 and H2. Whereas about 15% of African-American patients will have been infused with factor VIII that does not match their haplotype. And the major site at which there's a mismatch is this one amino acid, methionine in H1 and H2, and valine in some of the other haplotypes. So could these have something to do with inhibitor rates? Well, first of all, let's look and do some statistical analysis to see if the haplotypes correspond with inhibitor risk. Veal's study of 78 Black hemophilia subjects said yes. 3 subsequent studies from Schwartz et al., from Loken et al., and one from my lab, studying cohorts of up to 400 people, the latter one had 174 of those were African-American, found no correlation between haplotype and inhibitor risk. So a few years ago, the My Life, Our Future hemophilia database became available. I was one of a number of people that applied for access to this and got permission to look at it in order to attempt to address this question. So the database is a result of a collaboration between the hemophilia community, primarily. They're the ones who deserve our most of our thanks. Their respective clinics, the National Hemophilia Foundation, now NBDF, and BloodWorks Northwest Research Institute, and the American Thrombosis and Hemostasis Network, or ATHN. And through this program, over 6,000 hemophilia A subjects donated blood samples and data. So we will now proceed to describe the study that Dr. Ahmed primarily carried out the statistical analysis. I'll just briefly describe the databases that we use and that I presented to him and asked him to bring in his expertise as a professional statistician. So we had several databases that we combined to do this analysis. The first is the My Life, Our Future, which had a lot of data, but limited clinical data. We also were given access to the ATHN database, which had 4,677 subjects. We had the BloodWorks Northwest database, which consisted of the factor VIII gene sequences, which were sequenced at that institute. When we combined these 3 databases, and to make a cohort with data from all 3 of them, we had 4,169 subjects. And here's the breakdown: about 2,400 with severe hemophilia A and almost an equal number with mild plus moderate hemophilia A. And so we'll now have Dr. Ahmed, who will describe the analysis.
Thank you, Dr. Brad. So our strategy was, as in any hemophilia study, we divided our patient into 2 cohorts, depending on the clotting factor activity. Severe hemophilia A, which characterized by no circulation of factor VIII protein, with less than 1% of activity. Mild and moderate hemophilia A, which characterized by clotting activity 1 to 49%. Typically, these patients have innocence mutation or other small structural change. In each of the cohort, we conducted primary logistic regression to identify the factors associated with inhibitor development.
Dr. Ahmed, I'm so sorry to interrupt you, but we are having that distortion with the sound that we were concerned about earlier. Can we just do a little test with you again? Just say a few words.
Sure. So can you hear me now?
Oh, that is much better. That is much better. Thank you. Okay, I will get out of the way now. Thank you.
So we conducted binary logistic regression to identify the factor associated with inhibitor development. We adjusted our model for patient factors, age, race, ethnicity, Hispanic, non-Hispanic, factor VIII treatment regimen, prophylactic versus on-demand, factor VIII phenotypes, non-synonymous steps, age, geographic region, factor VIII clotting activity, and this was done for mild, moderate, and severe cohort. We also conducted some group analysis to compare the prevalence of inhibitor across patient factors and factor VIII mutation, genetic mutation, and the causes. So here we looked at the distribution of causing genetic mutation. And as you could see in the graph here on the left, almost half of severe hemophilia A caused by internet 22 inversion mutation, while mild and moderate hemophilia A mostly caused by missense mutation. The distribution of causal genetic mutation represent the US hemophilia A population. This is a distribution of ethnic groups. And as you could see from these several plots, that hemophilia A typically affects non-Hispanic white. Hispanic white represent 10%, non-Hispanic Black represent about 13%. These proportion provide us sufficient power to test our hypothesis. In severe hemophilia A cohort, we investigated the association between factor VIII mutation type and risk of inhibitor. Then we looked into large structural change and how it affects the inhibitor, and we found no association as it compared to internet 22 inversion. And this is a novel finding as a meta-analysis study was conducted in 2012, found positive association between large structural change and inhibitor development. This finding may require more investigation to replicate our finding. We also found that missense, frame shift, other mutation were associated with lower inhibitor risk as compared to internet 22 inversion. These findings were consistent with previous studies. In severe hemophilia A cohort, we looked into the association between ethnic groups and inhibitor development. So we found that Hispanic white had 88% higher odds of inhibitor as compared to non-Hispanic white. Non-Hispanic Black associated with 65% of inhibitor, 65% of odds of inhibitor as compared to a non-Hispanic white. We found no association with haplotype. In patients with severe hemophilia A cohort, we looked into the association between factor VIII replacement therapy, and we found that prophylactic only was associated with 40% lower odds of inhibitor as compared to both prophylactic and on-demand. Recent year were associated with higher odds of inhibitor as compared to years before 1965. This is the first plot, which shows all what we talked about. And as you can see here, on the left, you see the potential risk factor. in the middle, you see the vertical line. We also see the horizontal line, which represent the confidence limits. So the confidence limit here crosses this vertical line. It means no association or similar risk. And as you could see, for large structural change, as compared to uterine version, this is similar risk as the confidence limit for large structural change versus the vertical line. And also we see missense mutation, frame shift, and other mutation to the left of the vertical line, which is indicate that lower risk as compared to interim 22 inversion. And here, when we look into the ethnic groups, as we could see, the confidence limits for Hispanic white and non-Hispanic black to the right of the vertical line, which is indicating increased risk. For factor 8 haplotype, we see confidence limit cross the vertical line, so no association. For reflected only, we see the confidence limit to the left of the vertical line, and that means significantly lower risk as compared to both prophylactic and on demand. And we see here recent years associated with increased risk. It is not clear whether this is a true increase or could be due to frequent screening. We see here for geographic region no association. Here we look into two groups, high titer inhibitor patient and never inhibitor patient. We did not observe association, and this could be due to the small sample size. So this finding could be replicated in the future study using a large cohort of these patients. We also look into mild and moderate hemophilia A cohort. We divided the patient into 3 groups according to clotting activity level, 1 to 5%, a second group 6 to 15%, and the third group 16 to 49%. We found that low activity level, 1 to 5%, was associated with higher odds of inhibitor as compared to 6 to 15% and 16 to 49%. This could be due to the frequent bleeding, which requires more intense exposure to factor 8. The overall risk of inhibitor seemed to be lower in mild and moderate cohort than in severe cohort. So we also here assess the sensitivity of the association between ethnic groups and inhibitor development in different cohorts, and also use different models. We conducted a model without adjustment for factor 8 replacement therapy. We compared prophylactic versus on demand. We also considered a cohort of patients with interim 22 inversion. We looked into the severe cohort and combined interim 1 inversion with other structural change. We looked also into like severe hemophilia A and interim 22 inversion, and we compared large structural change and nonsense mutation, severe hemophilia A, and we stratified the cohort into missense cohort into factor 8 heavy chain and light chain. And lastly, we looked into mild, moderate cohort, and we stratified the missense cohort into factor 8 heavy chain and light chain. In all of these model and cohort, we found consistent result regarding the association between ethnic groups and inhibitor development. Thank you. Dr. Pratt.
Okay, thank you, Dr. Ackwood. So it's to me now to summarize a bit and put some of this into context. So our summary and conclusions at this point. Intron 22 inversions showed a similar inhibitor risk profile to all other combinations of large structural changes in the factor VIII gene. And this has some implications because there's a fairly prevalent assumption in the field that based on an earlier meta-analysis primarily and other smaller studies, that patients with an intron 22 inversion are at lower risk of an inhibitor compared to large structural changes. And this, the My Life, Our Future is the largest database that has yet looked at this, and we saw no difference. So there is really not strong, well, we didn't see any evidence in this study for partial tolerance that has also been hypothesized, partial tolerance to factor VIII as a result of the intron 22 inversion mutation. Racial ethnic differences in severe hemophilia A inhibitor risk were seen consistently in using multiple models, multiple subgroups of different size cohorts. We saw, as is already well acknowledged in the field, the prophylactic factor VIII therapy in this large cohort was associated with lower inhibitor risk. So we are left with a strong association between inhibitor development or increased risk of inhibitor development in the African American and Hispanic American population, and we still don't have a good reason for that. That's the bottom line. So we do need to not only further explore the basis of this, but we also badly need new therapies to promote immune tolerance to therapeutic proteins, factor VIII, and this is an issue in many other therapeutic proteins. So if you want some further reading, background reading in some of the issues that we looked into here, in particular, there are a couple of other publications out looking at the role of these non-synonymous SNPs, which is the same thing as amino acid variants, and inhibitor risk. So our group put out a study preceding the My Life, Our Future study of about 400 patients. So you could look that up in the reference cited here. And then also my group joined with Ken Mann and his group looking at that same group of patients, that same cohort of about 400 patients. And his group had developed a nice Luminex assay to characterize antibodies against factor VIII. And we saw that the antibodies did not bind differentially to recombinant proteins corresponding to the different haplotypes. So future directions, still looking for the basis for the unusual immunogenicity of factor VIII. This is a very active research area in numerous groups. Every year there are more interesting and provocative papers that come out. So just Google or go to PubMed, and there's something new every year. I want to emphasize again that everything we're learning about hemophilic immune responses to therapeutic factor VIII has potential applicability to other responses to other therapeutic proteins. In particular, a number of the methodologies that have been developed to look at these immune responses can be applied. And so there's wider applicability even outside the bleeding disorders community as well. The results that were in particular, the studies of factor VIII immunogenicity and potential tolerogenic therapies are relevant to analysis of new therapeutic factor VIII products, including those delivered via gene therapy. So again, I know I'm repeating myself, but there is compelling need for new approaches to promote peripheral immune tolerance to factor VIII and other similar protein drugs. So with that, there are a number of people to acknowledge from this study. So we couldn't have done any of this without the My Life, Our Future program. I'll start acknowledging local colleagues, the number one being Dr. Ahmed here, who I think did a wonderful job of this study. Other members of my lab have been involved in relevant studies of factor VIII immunogenicity, Drs. Pujavir, David Gunasekara, and others listed here. The project was funded by an NIH R01, by an earlier PATH study that was also funded by the NIH, by something called the Collaborative Health Sciences Initiative Program at CHIRP, and by UHS intramural funds. At the University of Washington, Drs. Jill Johnson and Shelley Fletcher have been very helpful with the My Life, Our Future associated analysis, providing data. Shelley Fletcher carried out or oversaw and carried out a lot of the factor VIII sequencing and was helpful with database cleaning as well. University of Vermont, Drs. Butanis and Ken Mann did the study that I briefly mentioned of antibodies. Biogen, Dr. Rob Peters and Seo Tan were valuable colleagues and collaborators. Dr. Glenn Pierce is a heroic physician scientist who helps all of us with our research. But so I thank the My Life, Our Future program, Athens Network, NHF, Bloodworks, Northwest Research Institute, and most of all the blood donors and clinical staff whose contributions make these studies possible. So we'd be happy to take questions. We can't see the chat due to the way our screen is showing, so please let us know if there are questions for either or both of us.
Super. Thank you very much, Drs. Ahmed and Pratt. That was really a very interesting explanation of your research and the many things that we can learn from this data that's really come from the contributions of so many community members. And I think it's really meaningful to bring this full circle and bring this information back to the community. So thank you so much for that. We will proceed now with our live Q&A. So if you have a question for our guest today, please enter it into the Q&A feature at the bottom of your screen, and we will do our best to answer all of your questions. Maybe we can start with a sort of a basic question from the introduction. Dr. Pratt, you talked about the possibility of infusing mismatched factor VIII. Could you explain a little bit what you mean by that?
Sure. So by mismatched, I'm talking about a factor VIII product that differs by one or more amino acids compared to the factor VIII gene in the patient. So it's actually, I described it in terms of an epitope. It turns into kind of a messy question, because if you have severe hemophilia A, what does it matter what the sequence is, right? And so I don't want to get too deeply into the weeds here, but potentially, even potentially, it's been hypothesized that even severe hemophilia A patients may have partial tolerance to factor VIII due to one of two possibilities. One would be some expression of the protein in the thymus early in development that is not that doesn't end up being factor VIII circulating in your blood, but that can influence the development of an immune response. But the primary hypothesis really behind the question of mismatches is that if intron 22 inversion patients circulate or express low levels of the protein, then even though they don't have any clotting activity, a sequence mismatch might be more likely because they're already tolerized most of the protein. Now we have another paper by Gunasekar et al. In my lab from just last year showing that intron 22 inversion patients actually respond to multiple epitopes. And then the study we just described statistically doesn't show a difference in immune responses of patients with an inversion compared to other large structural changes. So my conclusion at this point, on the basis of both statistical analysis and our attempts to find evidence of T-cell responses to these mismatch regions, is that they may occur very occasionally, but at such a low rate that they're not showing up as being statistically significant.
Right. Okay. That's interesting. It's quite complex. I guess maybe a more of a philosophical question. Way back when I was at university and learning these things, we learned about a normal sequence or a wild type sequence for a gene. And do you have any comment on maybe how we think about that now? The importance of how we decide what a normal sequence is and whether that is where the data comes from to term something as normal.
Yeah, I avoid the word normal when we're comparing genetic variants that are just that, variants. because there's the great risk that, like in a study like this, that you would say, oh, well, H1 is the most prevalent because we assayed more white people, and so the reference is white. Nobody likes that. But the fact is, the therapeutic factor VIII products out there, because the gene sequence was first probably determined from a white individual, happens to be the one that's most prevalent in white. It's also quite prevalent in the Black population, but these variants, to my knowledge, don't affect any aspect of factor VIII levels or factor VIII activity. It's likely that there will be some variants out there that affect. Well, we know about ones that affect activity to the point of causing mild hemophilia A. There are some, actually, I'm remembering now from Pete Lawler's group and some others, there are some that probably affect secretion and levels, like the factor IX Padua variant, for example, that causes, I don't know which one you want to call normal. I mean, the Padua variant is now used in gene therapy because you want the higher expression rate, even though was a rare variant, it's now used therapeutically. So I avoid the word normal.
Yeah, no, that sounds.
Unless
like
you're talking about something that causes a pathology.
Right. Okay. Makes sense. Thank you. Maybe a bit of a more technical question. So with 3 different databases being used for this project, was there any overlap of presentation of patients? Any chance of sort of duplication there?
There were a few duplicates, and those were resolved during data cleaning, which was quite a process, I can tell you. I did a lot of it myself. But the Athen coordinated with My Life, Our Future. I wasn't involved in constructing these databases, but we definitely do not have overlap, and we know that because of a separate analysis where we're looking at, which we're doing now, where we're looking at the whole genomes. So if you have the whole genome sequence of an individual, you absolutely know who's who. So based on data that I didn't present today, because it's just data that we're working with right now, we did not have overlap.
Perfect. Thank you. We've had a comment and a question come in. Thank you so much for the great lecture. Have you had any chance to compare this data with data from outside of the US?
Not yet, and I think that would be very worthwhile doing. Yeah, that would be of great interest. I think there are some good sized studies, cohorts in Brazil and several other places. That would be another whole study, and I agree it would be very worthwhile. We have not done that.
Well, and speaking of other studies, so if I understand correctly, there does appear to be a greater risk for inhibitors for people of certain ethnicities or races, but we really still don't know why. Would you like to speculate about what reason there might be, or what would be the study that would allow us to finally answer that question?
You want to answer any of these? We're waiting for a statistics question from my colleague here. I'm sorry, can you repeat the question quickly?
Well, it's so interesting to see that there really does seem to be a difference in the inhibitor risk for different race or different ethnicity, but we don't know why. It doesn't seem to be the haplotype, right? So would you like to speculate on what it could be? Or if that's, maybe asking a bit too much, what kind of study, how would you design a study that maybe could answer that? If you had any study, you could do all the resources, all the data access you need, what could help us answer that?
Yeah, so a family history of inhibitors is a known risk factor. And we didn't look at that specifically here. I still have questions about to what extent we're allowed to look at ancestry in some of the whole genome sequence data. Okay, it's totally okay to use race, ethnicity, self-identified, which is what we did with this study. That was data that was entered on a questionnaire. But earlier studies and probably upcoming GWAS and other studies of the whole genome sequences clearly show that there's associations in families, regardless of race, ethnicity, or whatever. If you have one sibling that develops an inhibitor, you're going to be monitoring the other children carefully, certainly during their initial factor VIII infusions, which is when you're most likely to be developing the immune response. So there's something else going on genetically. Lessard et al. Published an analysis of, I think it was most of my life, our future whole genome sequences, and found a couple of hits on proteins that were not previously associated. So that may be, well, I couldn't really interpret those. I didn't feel like I came away with an immunological explanation. It's a legitimate hit they got in their GWAS, but I didn't get a real explanation. We've carried out a GWAS, another collaborator here in my institution, not published yet, and got one or two hits, but the expert doing that said that, you know, sometimes these just kind of pop up and don't really mean much, and it wasn't anything that was that informative. It wasn't like a cluster of variants that pointed toward immunological pathways. So it's going to be a tough question. It's really going to be a tough question. And so we do need both types of research. One, just looking for genetic associations and so, but genetic associations will tell you who's at higher risk. They don't tell you what to do about it. So that's why we absolutely need to be funding research that deals with deleterious immune responses, and research into autoimmune disorders, as well as vaccination studies. You know, all kinds of wanted and unwanted immune responses are yielding more and more information about mechanisms, and it may be something entirely outside of hemophilia that gives us the clues as to what might be more effective in either preventing or reversing inhibitor responses. I think there's a lot of promise in the role of Tregs. It would be nice if we could do something simpler than some of the very interesting studies, some of which I participate in. And hi, David, if you're out there, to you know, engineer the Tregs of the patients. That'll be wonderful if it works. It won't be easily available to the entire hemophilia community worldwide. So ideally, you would want something that's much less expensive, much easier to administer, whether it has to do with initial exposures to factor VIII or therapies. It does seem that gene therapy, patients receiving well, it's mostly animal studies, sorry, but the animal model studies, the animals are not developing inhibitors at the same rate. So maybe the ongoing exposure to non-fluctuating levels of factor VIII is tolerated more easily by the immune system. And that's not an advertisement to go out and get gene therapy. There are many other factors that come into that, but that's an interesting observation.
Yeah, certainly. Very interesting.
There's some other possibilities that would allow ongoing exposure to factor VIII. Roland Herzog, a number of years ago, you know, developed oral tolerance models, and now expresses factor VIII in lettuce. And you can, the idea is make little pills of factor VIII and keep eating it, and that could be very useful. It may be enough to do that just in childhood. Maybe you would want to do that in an ongoing manner. But if you have ongoing, this is known in immunology, if you have ongoing low-level exposure to a particular antigen, that's much more tolerance promoting rather than being, you know, avoiding exposures for a long time and then getting a bolus of the antigen. We all know what happens in the springtime when the trees start blooming, right?
Yes, only too well. But it's very interesting, because that does also relate to questions around, you know, do you put people on prophylaxis? Do you use alternatives such as a bispecific antibody, and then they only encounter the epitopes when in a case of trauma or surgery, and then it's quite a large amount. So, you know, I think you have sort of spoken to that, but we have had a question asking, you know, how can patients use this data in conversations now with their healthcare providers? Is there a question they should be asking when it comes to their inhibitor risk, or how can they bring this into the conversations that they're having?
Yeah, I'd say, you know, we're looking for the mechanism, and first of all, we look just to see if there is a racial ethnic difference, and we had a large database that we could look at, so it's clear that it's there. I'm not a clinician, so I really try not to speak for I'm also not a patient. Physicians, I think, already had a good sense that African-American patients might be at higher risk. This study showed a strong association with Hispanic patients as well, so make sure you're monitoring them closely. But of course, I know you're wonderful people and you're monitoring everybody as well as you can already. Besides that, if get your factor VIII sequenced and you find out you have the haplotypes H3, H4, or H5, I wouldn't worry about it too much. That's one thing I would definitely have as a takeaway from this specific study.
That's interesting. That's a reassuring point.
Yeah, and it doesn't mean you're at lower risk. It just means you're at probably the same risk as others.
Yeah, it's probably not going to make a difference. Yeah. And I guess this is a little bit of an extension of the earlier question, but is there potential for this to impact health insurance and access to inhibitor care?
That's a very good question, and some of the clinicians could answer that. Shannon Meeks, if you're out there, you might want to speak up, because I know Dr. Meeks is an advocate of administering factor VIII together with emicizumab or whatever the next bispecific antibody will be called, just to maintain, like we were just talking about, the fairly consistent low-level exposure to factor VIII that would promote ongoing tolerance. Now you have to convince the insurance companies that they should be funding both the bispecific antibody and this very expensive protein factor VIII in order to avoid inhibitors. I hope the outcome will be that at least during the initial 150 infusions, which is the highest risk time in which patients might develop an inhibitor, I hope that they will be feel that the insurance companies will agree to fund that. Then you got to talk to the patients about whether, how are you going to get these infusions? You went to it's a complex question. You went to the bispecific antibody largely to avoid multiple IV infusions of factor VIII. So could you get it orally? Could you get it in another means? Ideally, you would be able to administer factor VIII at a low level consistently without requiring all these infusions. And I think that would impact the risk of developing an inhibitor, even for patients that are entirely on the bispecific antibody therapy.
Great, thank you. I really appreciate a lot of thought about what this means for people. I did want to come back with a comment from the original question, just suggesting that it would be really interesting to compare this data also with North African countries and the Middle East, given the prevalence of severe cases there. So I'm sure that's not lost on you.
No, not at all. And I sure hope those studies are funded, and we'd be interested in either reading about it when somebody else does it. It'd be even better if the scientists there were able to do it. So I'm looking forward to reading about it and would be very supportive whether my lab is directly involved or not. I completely agree.
Fantastic, super. Well, that wraps up the questions that we've received from our audience. I apologize to Dr. Ahmed that I did not have any difficult statistical questions for him. I'm so sorry.
Not even an easy one.
Not even an easy one, you're right. But we really do appreciate you both, Dr. Anwar Ahmed and Dr. Kathleen Pratt, for sharing your expertise and your time with this afternoon. I think we really all learned a lot about a topic that is really dear to our hearts. So it was really great to have such a detailed analysis and presentation and really solid data to help us with these difficult decisions. I'd like to also thank everybody in our audience for joining us this afternoon. This recorded webinar will be available on the NBDF website shortly after the webinar. Please note that the NBDF website has recently changed from hemophilia.org to bleeding.org. Hemophilia.org will still redirect you to bleeding.org, but you may want to update your bookmarks to this new address. If you go to the NBDF website under the Events tab and select Past Events, you will find all the recordings of past Wednesday webinars there. And you will also find under the Events tab upcoming events, including the next Wednesday webinars. So join us again on Wednesday, the 22nd of May, to learn about Own Your Path, which is a dynamic app-based program designed for adults with hemophilia A or B or von Willebrand disease who are on prophylaxis, and was developed through a cooperative agreement between NBDF and the CDC. Thank you so much for joining us this afternoon. Have an excellent week.
Thank you all.